Hello, Professor Hagee here back again. And in this recording, I wanted to demonstrate how I use a website called Nearpod to build my student lessons. And Nearpod is, again, a web-based site where you can add and embed multiple things into a PowerPoint or Google Slides presentation. And then it gets saved onto their site. And then you can push it out in a variety of different ways. It's either a student-paced lesson or do a live lesson within the classroom, or you're projecting the presentation onto the students' laptops or iPads or electronic devices. So I'm going to just kind of show you how I would build a lesson from this is I'm in Google, my Google Drive, and I've opened up, actually this is a PowerPoint, but it'll be in a Google Doc of a lesson on ecology. And so it's just a slideshow. And so the first thing I would need to do is open up my Nearpod extension. So if you're not familiar with their extensions, you can search and find different extensions. So if I wanted to get add an extension here, and if do a search. So if I wanted Nearpod extension, I just search Nearpod, and then you can add that, install that extension. Okay, so then it shows up under my extensions, Nearpod. Then when I click here, it'll open up a sidebar here with all the Nearpod things I can add in. Okay, so you can have drop and drag, audio clips, videos from the BBC, draw it, field trips. Some of these I've never used field trips. I've never used that. Some of these I've used the matching pairs quite a bit, and then of course polls, just short, just a question poll questions, quizzes I've used, and of course videos, and I use the YouTube. I'm sorry, add videos from YouTube by using this. I'll demonstrate how to do that. So let's say I want to add something into my lesson. So let's say I want to, let's say I want to just find a video, just a general video on, let's say, ecology. So let's say I want to do a little YouTube search. So click on videos. Now they do have some pre-made videos that you can search for their video library, but I want to go to YouTube, so I'm going to click on YouTube. Oh, and then if you have videos from your library that you want to add, you could do that as well. But I want to go to YouTube. I'm just going to type in ecology. Let's see what comes up. Let's say I want to use crash course. Just overview on ecology. So, and again, I can preview that if I want to see if that's what I want to use or not.
Episodes of Crash Course Biology.
But let's say, yep, I want to use the crash course. So I just click save. Okay, so here's the first upload. Now, what you can do here is if you want to trim the video, you can edit. You can do some video editing. So, and I've done that a lot, where I've found a video where there's only a certain part of that video I want to use, and so I'll just trim it down to that part and save that. Now the other thing you can do, with this is you can, as you're going through the video, you can embed questions.
38 episodes of Crash Course
Biology. I go through it a while, and I decide, he talks about something.
Population ecologist study.
Saying pause, and then let's say I want to add an activity at the 2 minutes and 20-second mark. And so you can either embed an open-ended question, or you can embed a multiple choice question. And let me just demonstrate this. Okay. So this, I'm going to just put in what is ecology. Study it. Okay, and then I hit save. And so now at that time point, you'll have a question, and you can come back and edit it if you want.
Why populations grow or shrink over time, depending on where they
So
are.
if we
When two or more populations of different species live together, we call that a community.
So if we back up.
But if you get 1 million oxygen molecules together, the same principle applies to
So when
organisms.
you reach
As you
that
put
spot
individual organisms together, they can interact with each other in their environments to create something larger than the sum of its parts. And just as every organism has a hierarchy of biological systems,
come on.
ecological order. Like when a bunch of members of the species are together in a certain area and they interact pretty often, you've got a population. Population ecologists study.
I can't remember if it does it now. Yeah, it'll pause. And during the lesson, then during the Nearpod lesson, then that question will come up. Okay. So again, you can go through the video and
non-living parts of their environment,
questions
like
where
soil and
water. Right. Okay. So once I've done my trimming, if I want to trim, or added questions if I want to do that, then you hit save. Now, if you don't want to do those things, then you just go ahead and hit save, and then it'll upload it in there as a slide. Okay. And so give it a second, and it'll eventually pop up on this little picture here of that video. Once it's done, there we go. Okay, so now that's in your slideshow. Okay. So again, if you wanted to add something else, so you wanted to add a question or a multiple choice, then you would, let's say you want to do a little formative assessment, the little quiz or a question. Again, you just click on it, and you can add your questions. And you can add a timer if you want to give them just a certain amount of time to do that. So you can set a time limit on the questions, and then you can add however many answer choices you want, and so on. Then when you on the quizzes or any of these other activities, when you look at there's a report you'll see for each student that participated in the lesson, either as a live lesson or a student paced lesson. Then you'll get to see that if they completed that, and what they did, how many points they got in the quiz, and so on and so forth. And you also get to see that in the report, a report after they've done the lesson. In the live version, you actually get to see it real time. You can see if there's a quiz that they did, what they did as they're answering that quiz. All right, so let's say I'm done doing all the things, and then you can just add regular slides, just like if you wanted to add slides here, like I'll add like learning targets and things like that. All you got to do is just hit, click and add a slide, and you can type on your slides, just like because you're really kind of still working. We're working in the Google Slide document right now. And okay, so now when you are finished, then you can. Let me show you one more thing before we go. So notice that the slides that I have right now, you can't edit. I mean, if you could convert it to a draw thing, but you could do a draw, and that's kind of cool too. I've added the draw slides where when it comes to a slide, then you ask them to draw out something, and then it draws out. Then you can see everybody's drawing, in the live lessons and stuff too. On this one, that we put in from using one of the Nearpod options here, then you can come back and edit that, right? So you hit edit that slide, it'll open it up, and then you can edit it, either delete that video, pull in another video. Okay, all right. So when I'm done, I save and go to Nearpod, and then it'll open up in Nearpod, and then it's saving here, and it'll just take whatever it takes here, a few minutes or whatever it might be. Okay. So while I was doing that, though, I want to show you one of the lessons I've done and some of the things I do with the lessons. So let's just go to, so here's a bunch of them that I've done for biology. So let's do my chemistry unit. I break it up into two parts. I do a chemistry of life part 1, and then a chemistry, which is basically an inorganic chemistry, and on macromolecules. I'm sorry, on polar molecules and acid base and so on. So let me just show you how I build my interactive lessons using Nearpod. So we're just going to go to a preview of that. So in my lessons, I always try to start off with some type of phenomena that's going to introduce this. In this case, it's an overarching phenomenon. I'm trying to just introduce the topic, right? Then I also maybe will have instructional phenomena that are specific to a certain part of the lesson as well. So in this one, I just had a little demonstration put up. So it's 2 clear liquids, A and B, pour them together. And when they meet, they change color, right? Okay. So I show that, and then I zoom in. I'll move I'm doing it, I think, on my iPad. And so I'll eventually zoom in so they can see that. Okay, so then I repeat this a few times so they look at it. So then I've embedded just some questions. So I just want them to a few questions about what do they think is going on. So with your table partner, discuss what do you see happening, give some explanations for what you saw. And so when they do this as a live version, where there's a code that, and I'll show you this here when we get done, that you can set up and do a live version in the class. And then there's also, you can get a code that you can link into your student management system that can be a student-paced lesson. And I'll show you that here when we get done. And then I have another phenomenon. So okay, so this, open this up. It shows another phenomena. Okay. And again, I'll let that kind of roll a few times. Okay. And then once they watch that phenomena, and again, we do a little discussion of their answers before I go to this one. And then I'll ask another question. And of course, as you probably know, then I'm asking them to talk about what they think is happening. Of course, in the other video, obviously looking for things that come up like chemical reaction, chemical change, those types of things. So I'm searching for questions that I get and kind of, talk about that. I don't necessarily tell them exactly what's going on there yet, but we talk about possible explanations, right? And of course, I'm looking for chemical reaction. Then we have this one, which again, I ask what they think is going on, what's happening in the video, and then we'll talk about, is that a chemical reaction? It's chemical reaction, chemical change, and there are times that, depending on the video, that I may not answer those. We may just discuss them and then let them we'll discover the answer in the lesson later on. And then I'll refer back to those videos saying, well, remember the reaction, with the 2 clear liquids, when we start talking about chemical change versus physical change, and we talk about, well, what are signs of chemical, that there might have been a chemical change, what was a physical change, and so on. Now this I embedded just a little what I call street knowledge assessment. It's basically a little pretest. So just 8 questions, and again, this will show up. I'll be able to see how they answered these. When I do the kind of the live version. I should have opened up in the live version here. So we're just going to skip the quiz there. Then I go into my lesson, right? So it's my learning targets. and then we'll talk about just going into the lesson, having a little discussion. Then again, this should be review. So my biology students are all sophomores, and then at Louisville, we as freshmen they take physical science. So this is really just kind of a review of things they would have covered in physical science. But I do kind of focus on a few things that involve living organisms. So we kind of pause a little bit and talk about this, the mineral ions that are needed and used, and we kind of have a little group discussion, asking them, what do they know any uses for some of these. And the one of them I do kind of focus on a little bit is talking about iodine, because that's one that this to them would seem out of place, like iodine. And we talk about iodine as far as its function. Ask them, have they ever know anything about iodine, or have they used iodine? But anyway, I talk about the story of discovery of goiter because of iodine deficiency. So I go through that little story about it. They moved away from the coast. People living on the coast were getting the diet from the saltwater fish and things like that, where they were eating their iodine. But anyway, I talk about that and lead them to that. Then we talk about they don't, where if they do, they know where they're getting their iodine, from lead to iodized salt and so on. So anyway, so then again, I go through embedded videos, questions, as you can see, compounds, elements. So this is now at the end of that first part. I'm sorry, and into the first section of that lesson. Then this is a formative assessment. So I want to do a little check for understanding here. So a little formative assessment, 10 questions, right, based on what we talked about in that first part. Now the second part is an activity. So, and I've kind of incorporated that activity into here. So it's a classic one, drops of water on a penny, and you guys may have done this, where I just, give them these instructions, send them to the lab. I tell them I got pennies and eyedroppers, and their goal first thing they need to do is write down their prediction. How many drops of water they think they can drip on a penny until it runs off, and then I want them to go do it. And I give them the freedom just to use whatever eyedroppers. They can repeat it, see if they can beat their high number, and so on and so forth. And then ask them to make some notes again on here. What was their high, and then what things worked, that helped them to achieve that, right? Then I ask them to draw a picture of what a side view of the penny, as they're doing this. What does it look like? Okay, so once they get done, we kind of talk about again, who's the high, how many drops, talk about what techniques seem to work better, and so on. Okay. So then I go to here where I actually have some pictures of that activity. And so then we discuss about why is it doing what it's doing? Why, because so again, this is kind of the phenomena, but this phenomena is specifically tied to a concept, polar molecules, right? This is the phenomena that I use to kind of start to get into that discussion. And talk about, why is it piling up, and why it's like it's hanging over the edge. And so, we make some discussions, and again, I ask them, can they explain, those results? Why are we seeing that, right? And again, I'm looking in our discussion of that, I'm looking to see if anybody, usually that attraction, they talk about attractive forces that comes up. And this, and of course, the attracting forces are thinking that the water is attracting each other. And so eventually, maybe not necessarily the exact terms as far as cohesive forces and adhesive forces, but they do talk about maybe there's some type of attraction. That's what we're trying to get, right? So then I go into talking about polar molecules to explain what we saw with the penny, and so again, that phenomena was just to lead us into the discussion polar molecules and those hydrogen bonds of attraction between polar molecules, right? Okay, so then it gets into mixtures and salt. Now we have a little formative assessment here again after that section, right? So a little formative assessment. Okay, so now this is leading into a storyline lab or activity that I do with them. And so in the activity actually is going to works into the discovering acid bases and buffers really in that lab. That whole lab is going to involve acid bases and buffers. So I start off by showing a little travel and tourism of Dominica, which is an island that I've visited many times. I go diving and hiking there, and I've probably talked maybe to the students about them in the past. But anyway, I'd show a little travel and tourism video on the island of Dominica. Then I talk about that I go to there a lot and scuba dive and I hike, and so then I give them this little story about one of my trips, and I was out hiking in the rainforest, and I ran across this native Carib, and he invited me to sit down and want to know if I want to sit down and rest for a while and talk with him, and sure, I said of course, I'll sit down and talk with him, and then in our conversations, he's asking about where I'm from and what I do, and so telling him I live in the middle of the United States, and I'm a science teacher, and then I talked, and then hearing that I'm a science teacher, he said, well, maybe you can answer a question for me. So as I make this bush tea out of a periwinkle plant, and whenever I add lemon or lime juice, the bush tea turns kind of purple, right? It changes color. And so I think about it a lot. I tell the kids I think about it for a little bit, and I tell them that I kind of have an idea what might be causing it, but if you had any bush tea that I could take with me and bring back to the States, then I could test it and to verify if my thinking is correct, my predictions are correct. And so yes, he easily gives me some bush tea to take back, and so and I tell him and I have this jar, this little mason jar with this purplish bush tea in it, right? And so so then I asked them, based on their little story and stuff, starting to ask them questions. Do you have any idea what might be causing the change of colors when the lemon or lime is added? So then they let them discuss that within individually, write out what they think, then discuss within groups, and then we kind of start doing our class discussion. And what I'm trying to do is get them to, and it does come up, a lot of them talk about, maybe it's this or that, but eventually someone's going to talk about acid. Maybe it's an acid, maybe it has something to do with that. And so, and then our discussion, again, getting them, narrowing them into and getting them on the path I want them to go. And so then, you ask this question about, well, what do you think would happen if we had other substances, and do you have any ideas? So I may have them give me a list of maybe ideas of substances they may want to add, and I'll have some obviously pre-selected that I want to use, and so then we. I can't remember my next question is here. Yeah, so then I want them to. So we decide, okay, we're gonna get these different substances, and then we're gonna add a little bit of the substance to the bush tea, and to see what happens. So I want them to make a predict or hypothesize, and what they think will happen. And so here we have. This is actually, this picture is an older picture. I used to use this universal pH indicator, so it's really not bush tea. But now I started using red cabbage extract, which you can make real easy, and actually can buy the red cabbage powder from supply companies. And so I started using that, and it works really well. Plus, I was always worried about the universal pH indicator is just dissolved in an alcohol, the ethanol, I think, or maybe isopropyl. But I was always worried about somebody tasting it. The cabbage juice can still have a little bit of an odor, so because our comment about, how did you taste it? I said, yeah, I taste it. Says, well, it doesn't smell very good. So, well, it's been sitting for a while, so they won't want to necessarily taste it. Anyway, so we have this whole, it's a multiple day lab. It probably takes the key, because what they do is I have them, I have a sample, maybe 10 or 12 or whatever substances, some that I have, some that they suggested, and then they go around and within their table groups, and they'll add, bush tea, and then add drops, and then write down color changes. And so in the lab, they have to predict for each substance, do color change or no color change, or they can predict specific color changes if they want. And then once they do that, then we see it gets different colors, right? And so they ask them to organize based on similar colors, and then we talk about pH. Start getting into, they're seeing some connections between certain colors of things that are acids and certain things that people know that are base. So we talk about that. And so then once we kind of discuss that and kind of get on that path, then I have them go back in the lab and then actually test the pH of each of the substances they add, and then see what correlations they can make between the pH of the actual substances and the different color changes that they see in the bush tea, right? And then we talk about it, seeing that the substance is a pH indicator, and so then they do a little research. Are there other substances that could be pH indicators, and find actually quite a few. Then eventually they do a buffer. I do also a buffer activity with them in that lab as well. So they to investigate buffers, and they actually, the question we're trying to answer is Alka-Seltzer an antacid or buffer? Because we always know about taking Alka-Seltzer because they had an upset stomach, maybe the acid. So they find out that Alka-Seltzer is actually a buffer as well, an antacid and a buffer. So anyway, again, that's that Nearpod lesson where I'm incorporating phenomena, activities, investigatory activities, trying to kind of again fit that new science standards, or that project based kind of. Now this is one that there necessarily isn't a high school standard for this, but it's one that, and keep in mind that not all lessons necessarily will have a high school standard for that, or even a middle school standard. But there are just things that I always do, no matter what. This is one of the things I do. It's just like I also do, science tools and measuring measurements. That's one of our first activities. We do like two kind of labs involving that. So anyway, this video is getting very long, but I wanted to let me go back here. I wanted to show you. So when you're ready to build that live lesson, we're going to do a live lesson, then you'll just project a code. So it'll have a code, and so then and then I have a different code for each section. So I have multiple sections. I have 3 different codes. And so it's when it's time for that section, then they'll go to joinnearpod.com, and they'll enter that code, and then this will pop up on their screen. And this is this is one I'd use for cell structure and function. It's a really cool two amoebas an amoeba that phagocytizes 2 paramecium. So all I'll do is I show the video of this happening. And what's really cool, search and find that video. This amoeba eats 2 paramecium, and they kind of shorten it down, but they follow all the way through to where it takes them in like this, and then all once the paramecium start freaking out inside the amoeba, and then all once they become these 2 blobs that fuse together. So I show that video, and then we kind of talk about what they think's happening, right? But I don't discuss it until we get all the way down the road a few days. Eventually we get to talking about lysosomal digestion, the lysosome coming from the, being made in the Golgi. And so when we talk about lysosomal digestion, and I go, I see, does that look, I refer back to that video. And then there's sometimes I'll ask a kind of open question, and then they'll make that connection. But that's what's going on with the paramecium, right, and the amoeba. So yeah, so you can, again, you can build that student-paced lesson as well. On the student-paced lesson, you have an option if you don't want them to skip different things you have in there, even the videos, and you keep that on if you're going to allow them to skip through that. But then when they're done, there's going to be a report. And let me just show you an example of that reports. Let me go to, I've got my online micro for Iowa Western going here. Let me go into one of those. So I can just click here and view reports. And so it'll show that lesson, and then it'll show the reports. So I can click on here. I can't remember what all I had embedded. So this one, I had just one. Now it doesn't show the video, so I had several videos. But what's interesting, again, that it will not, when they're doing a student-paced lesson, it will not go through that video. They can't go on until they watch the video, which is kind of cool. Then I had at the very end, I had kind of a formative assessment of matching pairs activity. So again, it'll show if they did it. So then I've got someone that didn't do anything, so they won't get any points for watching that Nearpod. So I give them points for doing that. But anyway, so you get that report from the Nearpod. Okay. There's probably things, and there's probably questions you have, and there might be things I left off that I was going to talk about, but we can talk that a little bit later date. But the cost, that was the other thing. So there is a free version. I'll put a link for that when I email about this video. There is a free version, but it's limited as far as how much storage space you have and how many. And I do there's a couple paid versions. I do the one just the basic paid version, and it's not cheap. I see it's up to like $159 a year, $150 a year. But I started using this 3 or 4 years ago now, and I love it. I love it. I'll pay the price. Well, actually, I don't pay it. I requisition at the end of the year for an IT expenditure, and so I pay for it, but then I get reimbursed from the school. Some schools you can talk in for a site license, which would be great. So, okay, so again, this is probably going on forever now, so I better end here. You can email me if you have any questions. Thank you for watching.